Hi everybody, I'm the greatest living inventor, and today we are doing my invention, automatic virus vaccine. So I'll write it down. Automatic virus vaccine. Okay, so first I'll read the description. Making a vaccine against any virus without needing to understand its structure or DNA, based on principles of assembly programming language. Okay, so I'll just read it to you, and once in a while I'll explain. Automatic virus vaccine. Background. There is no cure for any virus yet, except inoculating you, giving you a vaccine in advance, before you got infected. So my invention can work either in advance or even after you got infected. It's also a cure. When researchers are developing vaccines today, they are analyzing the virus, trying to single out what the human immune system can recognize, and then implanting this part in a non-lethal virus, which they introduce to our body. Okay, so they have the bad virus. Okay, this is the bad virus, and they see that it has something typical, like for example it has a head, or it has a tail. So they take something typical that they find, they put it on a non-lethal virus. This is a non-lethal virus, and they put on the non-lethal virus this thing. Okay, this typical feature that our immune system can recognize. Or they put the tail. Okay, so and then implanting this part in a non-lethal virus. This is one option. This is another option, which they introduce to our body. They put this one or this one into our body, and then our immune system and is ready when the real virus comes. See, for example, Ebola vaccine. Okay, so an example for this is Ebola. Using a vesicular stomatitis virus, this is the good virus, or chimpanzee adenovirus. So in the Ebola vaccine virus, This one is called vesicular stomatitis. And this one is called chimpanzee adenovirus. Okay. So just one moment, I'll take off my shirt. Okay, these are non-lethal, this is lethal, and the Ebola, and using this they make a vaccine. Okay, so they introduce it to our body, and to our body, and then our immune system knows when it is ready when the real virus comes. When the real virus comes. This one, this is Ebola. Okay, this has two drawbacks, disadvantages. It's slow and cannot be automated and depends on sufficient expertise. To recognize, to know how to make this, you need to be an expert. And number two, if that specific part of the real virus changes, mutates, the vaccine is useless. Okay, if let's say there is a new mutation, and instead of a cowboy hat, it has a clown hat, then this is now the new Ebola. And this is the vaccine, and it doesn't recognize it anymore. Or if the tail is a little different, let's say the tail is like this now, then this doesn't work anymore. Okay? So any small change in the real virus and the vaccine is useless. And also you need to be expert to make this in the first place. Here, in my invention, I will use a different approach, which is like a generic algorithm, which you just need to apply to your specific virus, and it gets you a vaccine. It gives you a vaccine. Okay, basic premise. So I'll erase this and explain my basic premise. It doesn't erase very well. This will have to do. Basic premise. My solution depends on the fact that our body's natural immune system can recognize and fight viruses if they are slow enough not to be lethal. In parentheses, like the common flu, which is not lethal. Close parentheses. But when it faces an enemy like Ebola, for example, it's advancing too fast and is lethal. Okay, Ebola is lethal because it advances too fast. So our immune system doesn't have enough time to recognize it. The problem, viruses command our cell's photocopier, copy machine, to repeatedly produce a lot of copies of the virus code. Okay, so I'll just draw it. If this is our cell, okay, this is one cell in our body, and let's give it also eyes. So all this is a cell. And in the cell there is a photocopying machine. Okay, this is how the cell makes duplicates proteins, and okay, and also let's make it lower so it doesn't get in its eyes. Okay, so this is the photocopying machine, and oh, this time I forgot to give it the cover. So this is the photocopying machine. This is the button that you push to photocopy to make another copy, and this is the cover. So I hope this is the photocopying machine. So every cell has something like this inside. Of course, it doesn't copy photos, it copies proteins. The virus also is a protein. Okay, so viruses command our cells photocopier compilation to repeatedly produce a lot of copies of the virus code until the whole cell explodes. So the virus comes, it comes into here, and it sits here. And okay, maybe I'll do it in not in colors but in shapes. Okay, so let's say like before, the one that are rectangles are the dangerous ones, the lethal virus. So the lethal virus comes into here, and it knows how to push. It's like it has some way to push this. It has like it's not a hand, but it's like an instruction inside the virus that knows how to operate this copy machine, to make another copy of himself. He is standing on the glass of the photocopier, and here where the photocopies come out, it makes another photocopy of the virus, and another, until the whole cell explodes. Okay, so so this is our problem. This is how the virus kills the cell, the bad virus. It's like the virus is stuck on the machine and press repeat after each copy. Okay, this is the repeat. Okay, so the solution. Think of a virus like a computer code, which means machine code, which means assembly language. In some part of the virus, there is this command equivalent to repeat in Pascal, or go to in assembly, in BASIC, or jump in assembly. Okay, so this is like the metaphor, but more accurately, the virus is like a computer program, a short computer program. So I'll erase this. Let's see what time. Okay, 18 minutes. And I'll drink something. Okay. So virus as a computer I'll just write it without a headline. So in some part of the virus there is this command equivalent to repeat in Pascal. Or go to in a BASIC, or jump in assembly. So even if you never programmed in any of these languages, you understand that this is the hand of the virus that can reach out this instruction, this special instruction in this virus, in the bad virus that can reach out and push the repeat button, the photocopy button. Okay, so think of the virus like a computer code, which means machine code, which means assembly language. In some part of the virus there is this command. Now I'm saying there are many kinds of jump commands in assembly language, but as you will see, the beauty of my idea is that we don't need to understand the language. We need to change only the single command to something else. Okay, so it's easy to ruin a program. It's hard to make a program work, but it's much easier to ruin, to destroy a program, to make it not work. Okay, so now how it works. How does it work? Plain one, when we create a non-replicating version of the virus, when we when we succeed in destroying or ruining this hand of the virus that pushes the repeat button, it looks almost identical to the virus, to the original virus, except it will only replicate once and just float harmlessly inside the cell. With your permission, from now on I will just call this the good virus. Okay, so the good virus only doesn't have this hand that it can press repeat and make endless copies. So the good virus only replicates once. On the other hand, if we put many of them in the bloodstream, in the human bloodstream, the natural immune system have enough time to tackle them and develop resistance to the good virus, and in this way also to its almost identical twin, the bad virus. Okay, plain 2. The bad virus needs to stand in line both in entering the cell. So I'll just try to draw this for you. Okay, so let's say the bad virus comes. No, I'm sorry. We are talking now about the good virus. But the good virus in my system doesn't look completely different, like a circle or something. The good virus in my system, if the bad virus is a rectangle, and okay, this is the bad virus, and it has some hand that can operate this repeat button, then the good virus is exactly the same but without the hand. Okay, so this is the good virus. It just doesn't have the hand, but it has all the same. It looks the same, other than this small part. It has if this has a head, this has a head. If this has a tail, this has exactly the same tail, and exactly the same head, and so on. Okay, so plain 2, another aspect of the solution. The bad virus needs to stand in line, both in entering the cells and entering the photocopy machine. So if we are talking now about being inside the cell, Okay, and they are now inside the cell. Then and here is the photocopy machine. So this one has to stand in line because we put a lot of these guys, the good guys. Okay, this is good, and this is bad. Virus, good virus, bad virus. So, and this is the photocopy machine. So, when this one wants to go to the photocopy machine, he finds a lot of these guys standing, because we put in the bloodstream of the person a lot of these guys. So, that only replicate once. So, it has a delay. They obstruct him, they interfere. And another way that they interfere is when they enter the cell. Also, the bad virus wants to come in. Okay, the one with the hand. But there are good viruses that without the hand, that are also trying to get to the photocopy machine, and because we put a lot of the good guys, then the bad guys this bad guy has to wait a lot, because there are a lot of good guys. Okay, we put a lot of good guys. We inject to the person a lot of good guys, good viruses. Okay, the bad virus needs to stand in line, both in entering the cells and entering the photocopy machine. This slows it down considerably, again giving time to our immune system to come up with an antidote. Okay, now another issue. This one is called can't read the code. Okay, so like you would say, okay, if we knew exactly how the bad virus works, we can do this. But we cannot read the code. Okay, we can't read the code. So that doesn't matter. We are using the fact that viruses are short programs of code, about a few thousand lines long. A few thousand lines is a short program. If you ever got to program seriously, this is short. They are coded in 4 letters, A, C, G, and T. Okay, so it doesn't matter. Why? Because the virus, let's put here a question mark and here a exclamation mark. Why? Because the viruses are like a short program, and they are made of only 4 letters. Let's paint them like this is A, and this is T, and this is C, and this is G. These are some proteins, some chemical or biological blocks, which is like assembly language. Assembly language is coded in 16. Okay, assembly language has 16. So this is a genetic code. Letters. And the assembly code letters are also very few. They are 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. And that's it. And also A, B, C, D, E, F. A, B, C, D, E, F. So these are assembly. Code letters. Or characters, okay? So so assembly language code is coded in 16 hexadecimal letters. So imagine if we have a short assembly program with somewhere a jump hiding, and we want a jump hiding, and we want to ruin that jump, to destroy this jump. We want to ruin that specific line only, without ruining the rest of the program. So on our first trial or compilation, we go to the first letter and replace it. So let me erase this and draw you schematics of how it works. So you see the resemblance, few letters, and it's like a code. So in a line of code in assembly would be one of these. A line of code in the virus would be one of these. Okay, I'm simplifying. It would be like 2 of these, but and this will be 2 of these. But to ruin it, you just need to ruin 1. It's easy. It's much easier to ruin something than to build it and make it work. It's much easier to make it not work. So remember, we are looking for that one specific line or letter that is responsible for the hand that can touch the repeat button, that makes the virus bad, that makes the virus replicate quickly. Okay, so now we will draw the virus in the same way, but we will look at its inner structure. Okay, so the virus is this, and it has letters of code. This is the first one, the second one, third one, fourth one. Okay, these are the lines of code. This is line 1. It's like you have a program, and this is the first line. Line. Okay, so this is the first line, second line, third line, maybe I'll do it in a lower first line, second line, third line, fourth line, and so on. The virus has a few thousand lines, but I'll explain. So on our first trial or compilation, I'm giving the terms of also of the programming. So in programming, if you take a program and want to make it run, then you have to compile it. So never mind, it's like a word from the computer world. But in our case, in the biological world, it will be trial. First trial, we go to the first letter, we replace it. Let's say we replace it to 0. And if it already was 0, we replace it to 1. Then we run our code. We test our virus. Did it work? Great. If it didn't work, okay. On our next trial, we leave the first letter as it was and change only the second letter, and so on. Okay, so let's do this in colors. If we have let's say this is blue, and this is green, and this is the problematic code, the red one. Okay, let's say the red one is responsible for this hand. This is what makes the hand. Okay, we don't know this, but I'm telling you. So at first we try the first line. We change it okay, this is first trial. First trial, second trial, third trial, okay, so so this is the bad virus, and it has, we are trying the first line. So we are changing it from blue to something else, like green. And if it was already green, then blue, okay? And then we test the whole virus. And we see that it's still bad, okay? It still has this hand. We don't know that it's here. This is the critical one, that the red color will be in the third line, the third letter, but we think it makes the disease, okay? So it's not good. So we test it, for example, on mice or something, or even on cells. We can take cells out of the body and just try it on cells. We don't have even to kill mice. So then we try in the second trial. We have the same original virus. And remember, this is the dangerous part, this is the bad part. So we try this. We go to the first line. Oh, we saw that the first line doesn't help us. So we say, okay, the solution is not in the first line. This is not when we look for the answer, this is not the first line. Let's see about the second line. So the second line, we go to the second line and we say, okay, if it was, we change it. If it was blue, let's make it green, like here. If it was already green, let's make it blue, okay? Or some other color. So we make it blue. And we test the virus, and it still has this problem, okay? It's still bad. It still kills the cells. So we understand that the solution is not the second line also. We can erase this one. And then we continue. Then we continue and we go in our third trial. We take the original, the bad virus. Okay, which has this hand. It's bad, it's killing the cells. And we test, so we know it's not the first line, so we are not touching the first line. We leave it as it is. We know it's not the second line, so we leave this second line as it is. Now we try the third line. So the third line, so we come to the line and we change it. If it was something, we change it to blue. If it's already blue, we change it to green. Okay, so we change it in this case to blue. And once we make this, we are destroying the hand. We are destroying the bad part of the virus. It doesn't matter into what we change it, but it makes it a good virus. So it's a good virus. It's no longer bad. So we know that this is the solution. Go to the third line and change it to blue. And this is the solution, this is the good virus. Okay, now, there is always a chance that by chance, by accident, by pure chance, we created another command which is equivalent in its functionality to the original command. Let's say the red is the hand, the jump command, but also let's say another color, if you put here like I don't know, orange. If you put here orange, it's also a jump command. Or if you put it, it's even possible that if you put here blue, it also means in this specific case, it's okay. By chance, it's a small chance, but there is a chance that here blue also means jump command, another jump command. The red means one jump command and the blue means another jump command. So how do we solve this? Because in assembly language, for example, there are many jump commands. There are jump if 0, jump if bigger than something, jump if always jump, JMP. Let's say we replace one kind of jump command in assembly with another kind of jump command in assembly. We will know that's what happened if we changed and tried each individual command in the virus and it's still a bad virus. So if we went through all the, let's say the virus is 1,000 letters, 1,000 lines, and we made 1,000 trials, and we went and tried to replace every single line, every single letter in the virus, and the virus is still bad. So this is our, this is the extreme case, when the rare case that I described another kind of jump command. Sorry. So if we see that's what happened, we try another replacement scheme. For example, making them all 1. And if it's already 1, turn it instead to 2. So, okay, so if we see that the blue, changing it to blue, doesn't work, the replacing scheme here in this example was change it into blue, and if it's already change it into green, and if it's already green, change it into blue. Okay, this was what we described until now. But we can make another replacing scheme, another system, that we say if we see that it didn't work at all, then we understand that this blue-green is the problem. So let's say changing it to orange, okay, the fourth letter. We say we had A, C, G, and T. So let's say that T is the orange thing. And then we say, okay, so we make another system that we change it into orange. And then if it's already orange, then change it into, I don't know, blue or red. The red isn't always bad. It's bad when it's in this position. Okay, it's important. The red, it's not like it's always bad and every time we encounter red, it means it's bad. No, the red is like one of the letters. But if you put it in the sentence in the correct place, if you put it in the program in the bad place, then only in that place it functions like in the bad, making the bad hand. Okay, so we can make another scheme when we replace it with the orange, and then it will bypass this problem of possible. It's in rare occasions that it's possible to have two kinds of jump commands. Okay. So now that we understood this, I'll erase this. Let's see what's the time? Okay, 50 minutes, we need to hurry. So I'm finishing. How each trial or compilation is tested, run in a test environment. Remember how in chemistry there are two sides of the chemical equation, reaction, the reactants and the products. So in the same way, if we have a culture of cells, that's what I said, we don't need mice, the typical victims of the bad virus, and we put it in a dish, in a Petri dish, and we infect them with the potential good virus, our current compilation, the last one, the one that succeeded. We look what happens over time. If they all die quickly, then we did not hit the jump command. The virus is still bad. I'm describing how we make a trial. I said trial, but now I'm explaining how actually we make a trial. The virus is still bad. If we see that the cells are still alive, we got a good virus, and we go to our recipe and produce many copies of the good virus. The good virus has, and this is the cure, the automatic vaccine. Okay, now last paragraph. The good virus has two advantages. One, it's very similar to the original bad virus, so whatever part or parts of it our body recognizes, there is a very high chance it will recognize it in the bad virus. Okay, so it has the head, it has the tail, it has all the parts, except the hand. When we fight, so there's a very high chance it will recognize, that our body will recognize the bad virus when we fight the real thing. And two, the second advantage, it has to go through a very specific mutation in order to return to being a bad virus. A specific single place and a specific value. We need the red in the third place, for example. So that's it. So I hope it was clear, and I'll see you in my next invention, and thank you very much. Goodbye.